everyone. In this video, we're going to describe innate immunity. So when we think about the immune system and immune function, we can essentially break it down into innate immunity and adaptive immunity. And we're going to cover innate immunity in this video and adaptive immunity in the next couple of videos. But so I just want to kind of suggest the difference between them. Innate immunity is alternatively called nonspecific defenses, and it's basically a collection of methods that the body's able to defend against certain types of pathogens. And so it's important to note that innate immunity is the same regardless of the pathogen. So it's a non-specific response. In comparison, when we have the ability to recognize a specific virus or a specific bacteria or a specific pathogen, the body can then create basically an army of forces that will directly attack that specific pathogen. So innate immunity kind of only takes us so far. It has the ability to sort of function as a barrier. It wants to sort of limit or deny entry of pathogens into the body. And if it sees them sort of circulating, then it wants to gobble them up right away so that it doesn't progress. But if it does progress, then the specific defenses will kick in a little bit later on and we'll be able to offer some help to the problem. So there are 7 types of innate immunity that we're going to cover in this video, and some of them will be familiar and some of them will be new. So we'll just sort of work through them. So the first one we want to mention is physical barriers. So we have, if we think of the skin as a physical barrier for starters, we know that the skin is a bunch of cells that are connected by desmosomes. So it's a very, it's a waterproof covering. It's very solid in terms of not letting things pass through. Also, the outer layer of the skin is dead, so there's nothing really to feed on. In addition, there are secretions of sweat and oil, and sometimes the secretions can be acidic in nature, so it prevents any sort of pathogen from being able to stay connected to the skin. And the hair also prevents insects and other materials from contacting the surface. If we think about the inside, so like anything that is a tract in the body, so the respiratory tract, which we'll draw as an example, or the urinary tract, anything that's a sort of a passageway in the body, we want to similarly protect from the entry of pathogens. So in the respiratory tract, we can sweep the mucus towards the pharynx if it contains any pathogens. In the urinary tract, we can flush out through the urine anything that's pathogenic. And if we think about the stomach has the ability to digest proteins. The next type of immunity that we like to discuss is immunological surveillance, which we mentioned in the last video was performed by natural killer cells, or NK cells for short. So we're just going to talk a little bit about what happens here. So the natural killer cells are floating all the time, and if they recognize a pathogen, so usually it'll be some type of a cell or some structure where it has some marker on it that we'll call an antigen that tells it that it's not a body cell or a body structure. And so the natural killer cell will actually attack the pathogen. It secretes something called perforins. And the perforins, I like to think of it almost as like nails, but the nails are like inserted into the membrane of the pathogen in like a cylinder, so it creates a pore. And so basically when we generate all these pores in the membrane, it will cause free flow of water and it will cause the cell to rupture, so it kills the pathogen. So I just want to suggest that what's important about immunological surveillance is that it can kill the pathogen right then. But just like if you think about having an infestation of bugs or something like that, you see one, it doesn't mean that you really have seen all of them. And so it's important to remember that just because the natural killer cell can kill the pathogen right away, there's no way for these cells to know how many other pathogens of the same kind are already circulating throughout the body. The next type we'll consider is phagocytes. So phagocytes are basically sort of a generic term for cells that engulf, and there's a couple of different kinds, and we'll mention just a couple of them here. So let's take a look at our phagocyte that's going to engulf this bacteria, and then it's going to destroy it inside the cell. And when we think about nonspecific defenses, we want to think about 2 things: macrophages, which are circulating and will enter infected tissues when there's some type of pathogen that's determined to be present. And then we can think about macrophages, which are derived from monocytes. So remember that monocytes are one of the 5 types of white blood cells, and so they can morph into macrophages, and then the macrophages can either be free, able to float around in the blood and enter and leave peripheral tissues. And so if that's the case, then we want to sort of just highlight what happens here. So the macrophage will be in the capillary, and then it can actually squeeze out of the capillary. So it just sort of like almost like I don't know, like slime or something, like in between the capillary endothelial cells. It just sort of reaches itself through and then enters into the peripheral tissue. And then when there's an infection or something, certain chemicals will naturally attract the macrophages to the site, and that's called chemotaxis. The other option for the macrophages is that they could be fixed, meaning that they're in a peripheral tissue and they don't move around. And we've already seen some examples of this, and I don't know that we've called it this before, but we can just sort of make the connection here, which is that the Kupffer cells in the liver are fixed macrophages. So they basically, whenever any type of pathogen, or like especially if we think about like bacteria from the large intestine, are taken up into the portal system, as soon as they enter the sinusoids, the capillaries in the liver, the Kupffer cells are able to engulf the pathogens right there. And the other example that might be familiar and is a little bit further back is microglia, which is the cells that are found in the nervous system. The next example of a nonspecific defense is fever. And fever is, they're just like the cells of the body, there are opportune temperatures for which pathogens can survive. And so one of the things the body will try to do is raise the body temperature to kill off some pathogen that's present. So the important question is, how does it know to do that? So certain types of cells, like white blood cells, which are leukocytes, and macrophages, will release pyrogens. They're basically chemicals that will change the set point in the hypothalamus, so raise the body temperature to a higher set point. I think, though, that commonly pyrogens are now recognized to be cytokines, which are chemicals that we know to be present in the body. Next, we have the complement system. So complement, this is a set of 11 complement proteins. And basically, I think it's best to think of it very much like the clotting cascade, because it's something where you need activation. So like there's a couple different ways that you can get activation. And then once you have activation, you will, the cascade will occur, and then you'll eventually have the desired outcome. So first, let's talk a little bit about what can activate the cascade. So option 1 is you have an antibody. So the body naturally makes antibodies, and they can be circulating in the blood, which we mentioned in the last video. So an antibody just happens to bind to the pathogen, and then because it's bound to the pathogen, it will kind of seek out and bind to the complement protein. Another option is that these chemicals called lectins, which are just like chemicals that are made as part of the innate immunity, they bind to the pathogen, and then that binds the complement. Or we can have C3, which is one of the complement proteins, binds directly to the pathogen. And any one of these things will trigger the cascade of reactions. And eventually what we're going to do is form something called a membrane attack complex, or MAC. And actually, it's very similar to perforins, but instead this time we're inserting the actual complement proteins into the membrane of the pathogen. So one at a time, we just kind of add these proteins, and it makes a pore. And then once we have a bunch of pores in the cell, we'll cause the cell to rupture because of the free flow of water. So we're using a bunch of different mechanisms to kind of do the same thing a couple of different times. The next example is interferon. So interferon is specific to viruses. So let's say that we have a cell that's infected with a virus. So just to put this into perspective and make sure that we're all on the same page, a virus is something that is essentially non-living. It's usually DNA or RNA. It could be circular. It could be helical shaped. It could be single-stranded or double-stranded. So there's a lot of different options for viruses. But basically, it's just instructions on how to replicate. And so it can't do it without a host. It can't do it by itself. So a virus will insert itself into a host cell, so like a body cell, and it will use the cell's structures, so the nucleus, the endoplasmic reticulum, the Golgi, to make more of itself, and then eventually it will cause the cell to rupture, and the virus will travel throughout the body and it will infect more cells after we've made a bunch of copies of it. So some infected cells, not all, but some infected cells are also able to produce something called interferon. And so interferon is released by the infected cell, and it has the opportunity to communicate with nearby cells. So cells that are in the vicinity that have receptors for these interferons can, the interferon binds and it works by a second messenger. And it basically causes the non-infected cell to produce some proteins that will protect it against the virus, so that it doesn't succumb to the virus the way that the other cell that already had been infected was. So it's kind of an interesting little process. And the very last one we want to mention is inflammation. And inflammation is really review, because we've already covered this previously when we talked about tissue injury. So it would be commonly associated with tissue injury even in this context. So if you had some type of injury that caused, a break in the blood vessel or whatever, or just even into the connective tissue, we're going to get stimulation of mast cells, which will release granules of histamine and heparin. We know that histamine is responsible for causing increased capillary permeability, so we'll have inflammation for that reason. And then we'll also get, sort of in conjunction with inflammation, we'll actually get complement proteins that arrive to the site, and we'll also get some macrophages that, like we mentioned, will squeeze out of the capillary walls and we'll try to engulf any pathogens. So not new, just review for that one, but the rest of them maybe new, or maybe a combination of new and old. So that pretty much covers the innate immunity. And so these are just the ways that the body can try to protect itself sort of generically. But if the body needs any more of a specific response or needs a more aggressive management of a particular problem, we move on to the adaptive immunity, or the specific defenses. So that concludes this video. Thanks for watching.